Day 2, here we are. We're going to try to get through everything that was basically on your Unit 2 test. And like I said, if there's anything else I can add to it, I will. If not, then okay, whatever. But first things first, let's go ahead to Chapter 6. These chapters were pretty detailed just because there's just a lot of important terms and things that we looked at. And a lot of it should be reviewed for you in terms of from what we did in Bio Honors. Yeah? 2-210. No, it's 2-210. Anyway, so the first things we want to talk about are the differences between two major types of cells, right? The main distinctions that we have there are prokaryotes and eukaryotes. So we have prokaryotes and eukaryotes. Prokaryotes, actually the name itself, karya, it means like good kernel. It means like a good center. So eukaryotes and prokaryotes are kind of easy to remember because you carry eukaryotes around, so you're made up of these. The only real thing that's distinctive about these is that these are bacteria, which you should definitely remember. Like those are bacteria. I should also just say there are some major differences there. Does anybody know what some of the major differences are? Membranes, yeah. What else? And where? Yeah, you're right. Okay, so we'll say more condensed DNA. And we will say for prokaryotes, very loose. Oh boy, loose. We'll just go ahead and call it chromatin. Yeah. Is that a hiccup? Yeah. I'm sorry. So also the main thing would be that DNA, it's more condensed, it's more compact because this is contained within a nucleus, which means these guys, they have no nucleus, which is really important. It's the defining characteristic of prokaryotes and eukaryotes. There are basically, there's some other minor differences here. We usually will phrase it this way. We'll say these have membrane-bound organelles. And these, they don't have those. They're not membrane-bound. But the organelles that they do have are more limited, and I'd say things like ribosomes. These would be a really good example of something that's very much not a membrane-bound organelle, but it's still necessary because ribosomes are really responsible for assembling or making all the proteins that are found in a living cell. Doesn't matter if it's a prokaryote or eukaryote; they still have to make proteins. That's why those are there. Yeah, I will also just throw this out there. Prokaryotes always have a cell wall. Whereas eukaryotes, it's kind of, half a dozen of one, 6 of the other. Sometimes they do, sometimes they don't. But prokaryotes always have to. They don't have a whole lot of complexity, but they definitely have to have more protection because of that. So, do you guys have any questions on the difference between prokaryotes and eukaryotes? Okay. I'm just going to add this in there. I know we kind of already mentioned it, but these guys reproduce with binary fission. And binary fission is wildly important to them because it helps them reproduce really fast. And that's one of the distinctive characteristics of bacteria and really simple organisms is that they reproduce so quickly. Anyway, let's go ahead and hit up some cell organelles. Sound good? Do I need to wait? Delay? Yes, ma'am. Yeah, both of them would have ribosomes. But the prokaryotes would be limited to pretty much just having ribosomes. And they have a few plasmids, but they're not very complex. It's nothing to write home about. They're pretty simple. They're dumb. They're dumb. That's all there is. Can I hit the button? Yeah.
Can I just have all the sticky notes?
Sure.
Oh, cool.
So cell organelles. Big one, of course, is going to be the nucleus. You should know that's where the DNA is kept. But I'm going to go ahead and add a little thing here. There is another portion called the nucleolus. This is where most DNA is held. But also, it's really responsible for making. Does anybody remember? What do they actually make here? No? Yep, they make the ribosomes here. This is really important because if you understand how proteins are basically assembled in the cell, you're going to have a much better understanding of why these organelles even exist, because almost all of them play a role in the assembly of proteins. So we'll talk about the mitochondria. Okay. And remember, I'm trying to highlight things that I feel like you're probably more than likely going to see on the test. You with me? So if you have your Unit 2 study guides, not everything from that will show up. Of course, there will probably be a question that distinguishes prokaryotes from eukaryotes, which is why I'm giving you that information. For this stuff, I'm not going to go through every single organelle because there's some that I just don't feel like need to show up on the test. You with me? So it's like, these are highlighting important stuff for you. But anyway, mitochondria, their whole thing is this is the site of. Woo! Thank you. Thanks, Chris. It should be an S. The site of cell respiration, as long as it's aerobic. As you guys will remember, aerobic cell respiration is a different process than anaerobic cell respiration. Anaerobic cell respiration happens outside of the mitochondria. So both processes, both the Krebs cycle and the electron transport chain, all happen within the mitochondria here. It actually has a double membrane, which hopefully now makes more sense because that membrane, that inner membrane, is where a lot of that good electron transport chain action occurs. Yeah. I would like to talk about ribosomes. Ribosomes, this is where translation takes place. And try to recall that translation is basically when you're assembling a protein from RNA. So when we're turning RNA into protein, that's what's occurring. Ribosomes are found on the rough ER, which is the endoplasmic reticulum, but they can also be found free-floating, and that would just be in the cytoplasm. They're both still assembling proteins. It's just their location of where they're dropping off those assembled proteins would change. I don't think you need to worry about that so much. Yeah, I think you're good. All right, so some differences between these things, and this should be really easy for you. There are differences between plant and animal cells. Animal cells have a few different organelles than plants, of course. Animal cells only have a cell membrane. There is no cell wall. So I'm going to go ahead and say cell wall for plant. Animal cells are going to basically contain only mitochondria. Plants will contain both mitochondria and chloroplasts. And then there is a difference here. These guys have small vacuoles. These guys have a large central vacuole. So of the organelles, these are the ones I'm looking at. These are the ones that I feel like are super important right now. Doing good? I'm not delaying. I'm just reading. I've been repeating myself a lot today. In all my classes, I'm doing review as well, and just really repetitive, and it wears down my voice, and I just crack my sanity a little bit. I'm recording all of them. I'm going to pick the best session, but the truth is I don't have enough time to get through it all, so I have to go home today and re-record it. So, yeah. And I had mac and cheese and baked beans today. Yeah. Nope. All right. I'm going to hit the button. Yeah? No? Okay. So let's go ahead and talk about some Chapter 7 stuff. This was all about the cell membrane, which talked a lot about transport and how things move in and out of cells. I should, well, I guess the cell membrane kind of acts as a good piece of evidence about evolution of life, the earliest forms of life. And looking at the membranes that we have, you'll actually see there's a lot of similarities between the organelles we have, their membrane, and the exterior membrane, the plasma membrane that our cells actually have. They're so common, in fact, we think they're pretty much made of the same thing, which means they probably formed under the same conditions. So you should be familiar with the idea of endosymbiont theory. You might have heard this phrased as endosymbiosis, which is fine, but common cell membrane, endosymbiont, common cell membrane features show organelles likely formed as infoldings of cell membrane. As a matter of fact, they're even starting to, like, the whole theory by Dr. Margulis supports the idea by saying if you look at like the cell membranes of anything, especially eukaryotic cells, you will see that their organelles have the same structure. That's actually why vesicles work so well. Vesicles can merge with the outer layer of the cell membrane and form with it because they're made up of the same components, which now brings us, go ahead. Literally just an inward folding, and basically if my hand is a surface, it's going to fold inward, and then it can close off, and that makes an organelle. So an infolding is folding inward. Yeah. So what we have here are some important components. You should know that the phospholipid bilayer is what we call this thing, but we prefer using the fluid mosaic model. And you guys learn this a little bit different than kids would learn in, let's say, honors biology, because we talked about the fluid mosaic model, showing that it's not just the lipid bilayer that's playing a role. If we actually look at the components, it's roughly a balance about, like, I'd say about 50 to, let's say 50% would be about phospholipids. but about 40% would be proteins. That's just a rough estimate, but we're saying like it's almost equal parts proteins and lipids. And proteins have their own job. They reinforce things that are found in there. They can act as receptors. They're kind of like the functional unit of your body. Therefore, they're the functional unit of the membrane. But the phospholipid gives the fluidity. It gives a protective layer, a sheet, that separates it from the surrounding extracellular environment. So yeah, you guys know if you have like your phospholipids, and they look like this, right? We use a phrase to describe this region right here. This region is considered hydrophobic. And the region outside of that, that's what we call hydrophilic. And hydrophilic just means it loves water. Hydrophobic means it hates water. This is why the only thing that can pass through here freely are small, non-polar ions in most cases. And that's because since they're so small, they're much smaller than the lipids, so they can actually squeeze between them. They're non-polar because these tails that are found here, these are non-polar. And if you guys remember, non-polar is an equal sharing of electrons. So overall, if they share a similar charge, then they can diffuse through there. If we ever mention anything that was fat soluble or oil soluble or lipid soluble, that means it can diffuse inside of cells pretty readily. I know we're kind of taking some later concepts and applying it now, but that's kind of the point of this. If you guys remember hearing about like, remember the receptors that can be found inside of cells? Yeah. They could be on the outside, exterior, but they could be interior receptors. Those interior receptors mean something had to pass through the membrane. How did it get there? It might have been a non-polar substance that got through there, and that's going to convey that message. So it might be a primary messenger in that regard. Anyway, you guys feeling okay about this fluid mosaic model? Yeah? You need me to drag my feet? I'm just reading. Okay. You should be familiar with the concepts of diffusion. Yep. And I guess technically we call it simple diffusion, but whatever. Just remember that's the movement of particles from a high concentration to a low concentration. And you should be pretty familiar with something called facilitated diffusion. Facilitated diffusion is still diffusion. Like that's the key word there. It still goes from high concentration to low concentration, but it uses a protein to help transport through the membrane. Okay, so facilitated fusion is pretty big idea. A lot of things can pass through things that way, and they can be large, they can be small, but the point is they have help. They couldn't do it by themselves, so they still use a passive form of diffusion. And then we talked about the forms of, by the way, these are all called passive, aren't they? Oh, I'm assuming you just know this, but you should know about osmosis too. It's just diffusion, but it's diffusion of water molecules across a gradient instead of just stuff, instead of a solute. Does that make sense? Does anybody need me to review those two? I think it's pretty easy. So both of these, all three of these, sorry, are called passive transport. Which is awesome. It's good. It's consistent. It helps us survive, but there's a cooler one out there. Active transport, right? Okay. Active transport comes in a few different forms. We can separate them basically into two categories. We have endocytosis. We have exocytosis. Endocytosis, I think there's only 3 forms you need to be aware of. There's pinocytosis, phagocytosis, and then a tricky one.
You're doing a really great job, Mr. Olson. You're so good. Hang in there. You're like an Olson Receptor G. Hang in there, everybody.
So you got pinocytosis, phagocytosis, and receptor-mediated endocytosis. Pinocytosis is associated with drinking. Phagocytosis is associated with eating. And I don't mean like we do these things. What I mean is that the cell does these things. So drinking would be taking in really small molecules a lot all at once. Phagocytosis would be like I'm going to ingest one large molecule at a time. And that's usually associated with like your your white blood cells just actually ingesting things. But receptor-mediated endocytosis is kind of like this. So we're saying like you have these little receptors there. And as soon as the molecule attaches to this, it signals a response on the inside that says create this infolding. So it actually closes off and eventually seals it off so it can form a vesicle and bring that on the inside. So the body can say like, oh, I got the thing I'm looking for. Let me bring a lot of it all in at the same time. This would be pretty common in things like neurotransmitters. That would be the most common thing you'd probably find that in.
Those people are stuck in the ditch.
Sure are. Right. I get it.
Come on,
I'm reading. I'm not delaying. Sorry, it just takes me a long time to read. No problem. That's why I'm here. Thanks. I appreciate it. You're so nice. I know. All right. So let's talk about some of these terms here. Water potential.
Is this still chapter 7?
Sure is. Yeah. It just exits. There's nothing other than that. That's it. So it's just the reverse process. Things leave. I wish it was fancier than that. It's not. It's just cell poop. That's all it really is. So water potential. Yeah. Yeah. So the main thing you got to know for this is that water moves from high pressure potential to low pressure potential. I don't think I'm going to make you work any formulas. If you guys remember, there was some like, there's a formula for finding solute potential. Do you guys remember that stuff? Yeah. Do you remember the first formula, the simple one? No, the simplest formula was. What do you think? Yeah, so water potential is equal to 2 different potentials added together. That's all it is. So you have pressure and you have solute. So you just add them together. It should equal 1. and that's water potential. But that's basically it. So look at that just simple formula, and that's all you're looking for. So if I say, like, what's the number for this one? You should be able to tell me.
Wait, is that something you tried it?
Yeah, it's called psi. I believe the symbol is called psi. It stands for Poseidon, which was carrying a trident because he was the god of water and all that stuff. Anyway, so yeah, there you go. Hypertonic, hypotonic, isotonic. I don't know what that music is.
You do?
Yeah.
Is it the Dr. Dallinger was listening to, like, what is that music? The polka music, because she got an accordion today.
Wow. Could be polka music, yeah. All right, so I'm going to show you a thing I showed you a couple years ago, but I'm going to change some of the terminology, which now should all fit together. Right? So you have a cell inside of a watery solution. In a hypertonic solution, I've always told you guys this, it's goofy, but like if you're hyper, your parents tell you to go outside, right? So the higher concentration of the solute is found outside, and there's just a little bit on the inside. Hypotonic means there's a higher concentration of the stuff inside or underneath. That's what hypo means. An isotonic means it's a nice healthy balance inside the cell and outside the cell. So what we're saying here is water potential, and we're just going to like write this water potential. The water potential inside and outside will be different. Actually, I should not do water potential. I do solute potential. What am I doing? Why are you guys being this way, huh?
What do you mean this way?
Why are you being this way? So the solute potential shows that the solute wants to move in, but when it comes to water, water wants to move this way. So we're saying you have a lower water potential, solute potential here, and a higher water potential here. Okay, and water wants to move from a high concentration to a low concentration, so the solute potential is higher there. Go ahead. In this case, yeah. And just the opposite would be found here. I'm just going to write the psi symbol now. You'd have a lower there, higher here. So you have a higher concentration of water there. And then they'd be even. So they'd both be at 0. So they wouldn't change. Doing okay? Mm-hmm. Do I need to like delay? What's up? Yeah, I'm pretty tired. I'm pretty tired. No, just tired as in kind of fed up. Yeah. So, can I hit the button now? Yeah? No objections? All right. We're still on chapter 7, aren't we? No, we're not. Finally. Let's get out of this mess. Am I doing on time? Probably terrible. Yeah, it's awful. Let's talk about chapter 8. So we got energy. We called it Gibbs free energy, delta G, right? Remember, delta means change, and G represents energy. It's actually Gibbs free energy, but whatever, right? So there's that first law of thermodynamics. That first law basically says energy cannot be created or destroyed. So it's always going to be there. It can get repurposed, but it can't be created or destroyed. Second law says entropy is always increasing. Okay. So there's Gibbs free energy formula, which is delta G equals delta H minus T delta S. But for all of our purposes, I'm going to tell you, you should know this one. Oh, my bad. That's final. So this one right here, you should see either a positive or a negative result. If it's 0, you're not having any energy transfer at all. So it's like you have a closed system, and that system's not allowing any new source of energy. So it'll just stay static. It won't do anything. So we're saying if our final amount of energy is lower than our original here, we've had a loss of energy. So if you get a negative value, we call this, which phrase did we use? Yeah, okay. We said exergonic. Okay, so something less than 0. That's all it's got to be. If you're positive, we use the phrase energonic, which now relates us to the ideas of catabolic and anabolic. If you're breaking something down and releasing energy, you're probably catabolic. And if you're building something up, you're anabolic. Okay. So Gibbs free energy, well, it looks like a big, ugly, complicated formula. If you look at that bottom thing that I circled, if you end up with a negative value, that's just going to tell you that you have a catabolic process. You're going to be building something up. Or I'm sorry, breaking something down. Catabolic breaks it down. Anabolic builds it up. Okay. Any questions there? No? Anybody still need me to read? That's what she said. Thanks. Can I hit the button now? ATP. You should be pretty aware of this molecule. Adenosine triphosphate. Yes, still chapter A. Just remember, there are 3 phosphates that are attached to this molecule. And this third one's pretty unstable. So this thing will break off pretty readily. And if it does, we can see a lot of energy being released. Just remember, we got to add, if we have another molecule called ADP, if you're going to add a phosphate, which we'd call an inorganic phosphate, to do that, we have to use this process called phosphorylation. And this is how we transfer energy, essentially. This is actually not just used for ATP, it's used for other molecules as well. You guys also might remember, well, I'll wait for you. I need the space. Yeah, it's inorganic phosphate. Yeah, just adding that phosphate. I need a good nap and a coffee. I need a nap and then coffee and some gum.
Do you want some gum?
No, it'd be after the coffee. It'd be after the coffee because the gum, like coffee, tastes bad.
Because I have gum.
Yeah, I know. I'm saying like I would chew the gum after the coffee. Anyway, thanks for the offer. Gum.
Hearing.
Just on you in your backpack.
Like no, like I have cups of them, and then in Lowry's class he lets me make coffee.
That's very odd. But no, thank you. I'm good. Thanks though. There's this thing of energy, right, being involved on a graph. You should definitely be familiar with this. This is like that familiar shape that you should all know really well, right? And we talked about something called activation energy. And I remember on your test for this very test, a lot of you messed this stuff up. So you may want to pay attention. From here to here, like this is our starting point, right? So let's say, I don't know, let's say this is 0 on the graph. We're starting up here. From here to here, that's called, I believe it's A of E. Is that right? No, it's E of A. This is called E of A, which is activation energy. This is the amount of energy required from here to cause a chemical reaction, right? So when we were talking about enzymes, the whole job of an enzyme is to make sure that it lowers activation energy, so you use less of this stuff. So at this point, we're saying we can have the reaction using, oh, that's a W, using an enzyme. Okay, so you're going to lower the activation energy. You're using less energy to make the same reaction happen. Yeah, that's a question that was asked to you guys, and I know a good number of you messed that up. But anyway, things that can affect the ability for the enzyme to even work. Enzymes are affected by or changed by, I think we mentioned this yesterday, but the pH of the solution that it's found in, their concentration, and overall the temperature. Remember, the temperature can be too hot or it can be too cold. And that's because of the hydrogen bonds that have formed between the proteins. They have a particular structure, a particular shape. And if you change that, then you're going to change what that protein is. And in turn, your enzyme or your catalyst are not going to work properly. Yeah, you should also be able to tell me if you start up here and you end up here, let's say at a higher level, and if you end in a lower state, which one's catabolic and anabolic?
Wait, what was it?
I'll just remind me of this tomorrow.